I would like to thank NMIN for giving me the opportunity to talk about the PharmaCore, which, as Diana mentioned, is led by Drs. Marcel Bally and Star Lai. And my contact info is here if you need to get a hold of me. And so our mission, it's a very simple mission. It's to help provide support to NMIN investigators and research partners to facilitate the preclinical and clinical development of novel nanomedicines to meet regulatory requirements to advance new therapeutics from the bench to the clinic. Some areas of expertise that we have include formulation development, as well as characterization of biological activity in cell models and animal models, and as well as manufacturing. That's from bench batch to, so small scale to GMP manufacturing. We are located in Vancouver at the BC Cancer Agency's Research Institute, which you see here. And then you also see in the picture some of our staff doing various activities. So as novel medicines nanomedicines move through the drug discovery pathway from the bench to the clinic, there are several phases that occur as shown in this diagram. And these include discovery, which universities are very, very good at. And then proof of concept, preclinical studies, to first in human clinical studies, and there are 3 phases, and then regulatory approval. This diagram is a little bit misleading because you actually do need regulatory approval to move from preclinical to the clinical phase, which is a CTA or clinical trials application for Health Canada, or an investigational new drug IND application for FDA. And there are other similar ones for other regulatory bodies in the world. So PharmaCore specifically has capabilities in vitro and in vivo, which I'll talk to you about in future studies. And this culminates into performing safety pharmacology or good laboratory practice studies accredited by Standards Council of Canada. And this is essentially to test any type of toxicity and ensuring it's safe to go into humans, and could also provide some evidence of the first initial dose to use in humans. What I wanted to point out about this slide is, especially for nanomedicines, a lot of the other activities happen alongside this, which is formulation and analytical, how you're going to quantify your payload as well as your lipids in various solutions, such as just aqueous solutions and then tissues and blood. And as you go through the discovery pathway, you need to produce more and more material. And if some of you have gone through this, what's in a test tube is not necessarily, to scale it up, you might have some challenges and you might have to change your formulation of it. So, and so we're capable of going up to GMP manufacturing, which would help in early stage clinical trials. We are capable of about 10 liters, but that depends on vial size. To perform GMP and GLP, we have a quality management group, and they, I'll talk about that later, but they're very important to ensure that we adhere to any of the regulatory requirements needed. And unfortunately, that slide, it shows it as a linear process, but it's not really linear. So in this figure, it shows basically when you come up with an idea and you work with your technology development office and your partner to get some of these studies done, you have different go, no-go decisions. You are doing some of these things in parallel. You might be improving formulations along the way, doing your preclinical, and then also going on to do, if it looks promising, you've got enough proof of concept, you're going to move on to GLP toxicology and our manufacturing and obtain stability testing, and hopefully use that material for the GLP. And that will then form a batch record for your clinical trial. And we'll talk a little bit more about that. So in brief, I just wanted to talk about some of our in vitro capabilities. So we are oncology focused. That is our expertise, but we work with a lot of partners on other disease indications and so forth. So it's not limited to this. We do cytotoxicity, which is to assess the degree to which a substance can cause damage to a cell, ideally to cancer cells versus not in normal tissue. And so we can do the standard MTT, Alamar Blue, cell titer glow. But we also have an in-cell analyzer, which will directly assess live dead. This is done with fluorescent imaging and markers, and you can get actually nice pictures as well. We also use that to do combinations and see if there's synergistic combinations. And the in-cell can also do high content screening, which would look for other assays such as cell cycle studies, co-localization, apoptosis, and colony counting, and so forth. And we do other tissue culture assays, including the FACS and IncuSite, which is up there. It's a machine that sits in the incubator and assesses growth and can count the cells and so forth, and Western blotting, as well as others that we've done. For preclinical studies, ideally, this is started with a maximum tolerated dose defined in dose range finding studies. And also, I'm hoping that people use well-characterized formulations and that are tolerated in vivo. So a DMSO formulation is not tolerated. And so we might have to do some formulation at that point. Especially in the nanomedicine space, you may have to do additional pharmacokinetics in vivo to optimize your formulation. And this may include biodistribution, which assesses which tissues this is accumulating in, or whether it's accumulating in a tumor, for example. And you may do pharmacokinetic, pharmacodynamic studies assessing whether it knocks down mRNA or after a certain time point, or protein expression, and so forth. Further proof of principle studies include model development. So the model has to be reproducible and robust. And so it has to be in a strain like this. If we've done it in one strain, we might have to do another strain. So that should be done. It shouldn't be something that's skipped, because then you won't get data that you can use. We also do efficacy studies. These are examples of cancer models, but there are other models that we do that can be developed in-house. We've seen a resurgence of syngeneic tumor models, which are mouse or rat cells used in mouse or rat models. And that's because the immune system is intact. Xenograft using human cells, we can do orthotopic. So that's tissue being in their native state or native tissues. So a brain tumor would be a brain xenograft in the brain implanted. And we also work with Pancreas BC to do patient-derived xenografts, as well as others. So we're not limited here. Just wanted to briefly touch that we do have an IVUS machine, so we can do non-invasive live imaging. This is for both bioluminescence and fluorescence markers. And you can see the picture. We can also work with other groups in our building that can do different other modalities, imaging modalities, such as micro-PET CT. But the in vivo live cell imaging is great for orthotopic models, metastatic models, and we can work with you to identify the best marker and whether bioluminescence or fluorescence is best. And lastly, formal GLP studies are performed by our group. We are accredited by Standard Council of Canada, and we are audited every 2 years. This is a very SOP-driven, documentation-heavy exercise, but it really fulfills the requirements for your IND or CTA for rodent studies, or the best, you may have to do other larger models, and includes a repeat or multiple dose study and assessing acute, chronic, or subchronic toxicity. The endpoints are usually analysis of blood, so for complete blood cell counts or differentials, and as well as blood chemistry analysis. In addition, histopathology of multiple organs. And then this is all reviewed by a board certified vet pathologist, and a very big report. Just a brief history. Our name outside, we are known as the Investigational Drug Program outside of the NMIN, as we were established back in 1995 with initial funding and loans by the BC Ministry of Special Programs and Western Economic Diversification. This allowed the construction of a small clean room facility within the clinic, actually, the clinic side, the clinic building, and it's about 850 square feet. At the time, there's a lot of activity in manufacturing, so it was well used and reached maximum capacity by 1997. And to date, we've done over 125 successful parenteral batches. But it's higher. This is a bit of a dated slide, so it's a bit higher, and we've had a lot of interest recently with COVID and newer nanomedicines taking off and requiring this support. Just as historically, we have worked with a lot of nanomedicines in the past and currently, and these include nanomedicines with various payloads, such as cytotoxics, siRNA, mRNA, and small molecules. Some of these are now commercially available, but we would have done the initial stage for phase 1 clinical trial. And this is just an example of what it looks like in the clean room. We have 2 rooms, a prep room and a batching room, and allows for material to be made in sterile conditions. And just to touch on our GMP manufacturing capability or manufacturing scale-up ability. So it's ideal that you may start with a bench batch to establish a batch record. And then from there, you will move into the clean room. This allows, once it's manufactured, it will be vialed and labeled and then inspected to be released. And at this time, you can also do stability protocols and different temperature environments and time frames. And all this requires lots of documentation and quality management oversight. To support both GLP and GMP activities, we have storage and archiving facilities. These are monitored 24 hours a day. These are monitored for temperature and humidity, and allows for storage of all your inputs, your raw materials, and your API, as well as your final product, and quarantining of the final product until it's been released and met all the specs. I wanted to touch on quality management. Quality management has 2 arms, sort of, quality control and quality assurance. Quality control is an arm that approves and rejects the manufactured drug products. It oversees that the facilities are being maintained and as we've stated, and qualification and maintenance of all equipment and validation of testing methods. There's a documentation control system. They do the stability testing, reporting, and training for the GMP, GLP personnel, and implementation of SOPs and all the reports and protocols required. Quality assurance assess that we're conforming to our quality management system, and they conduct both internal and external audits and inspections, and will monitor for compliance with our SOPs and ensure that we are doing things according to OECD, Health Canada, and FDA regulations, as these can change over time. And I'm also involved in the archive, document control, and change control. I didn't touch on project management, but we do provide support for grants, and I provide quotes and so forth for both manufacturing and preclinical needs. And it could be just for a company budgets. Anyway, so with that, I'd like to thank our team members. We have several people who are, most of these people are all highly qualified personnel, all of them, and wear multiple hats in our group.
Thank you, Nancy. Fantastic presentation. And to everyone that's listening today, it is just such a privilege for us to have access to the breadth and depth of expertise that exists in what's been known for a long time as the investigational drug program and is known to us as PharmaCore. I think it'd be really great, Nancy, at some point if you could maybe even with one of the investigators share kind of a story like on a nanomedicine that you've worked on, all the way from maybe kind of early stage PKBD to some efficacy or whatever it might be. I know my research group has worked with your group, or we've used your group for GMP manufacturing and scale up, which has been tremendous because I don't have that expertise myself. But I think that could be really helpful to folks.
I agree. Yeah, there's a lot of things I couldn't tell because it is very helpful for everyone to contact us and maybe consult with Marcel and I. Like I often tell people to work backwards from efficacy. So you're not repeating steps, you're using the same strain that you'll end up using. Could be for your manuscript, but it could be for a bigger project.
Yeah.
And so things I couldn't bring up in the presentation, but
Sure.
things that you will learn and the ways you have to think about performing these studies and getting ready to for your product to move on.
That's great. Thank you.
Nancy, in your talk, when you were talking about preclinical studies in vivo, you said we prefer to work with well-characterized formulations. And so if you had any advice, sort of what are the top 3 tips that you would give to the people on the call today if they were thinking that they might want to work with you? How would you advise them to assess their state of readiness to approach you to leverage this incredible facility that you lead there?
So I mentioned that we wanted well-characterized formulations. So in the nanomedicine space, assessing that you don't have big and small particles are very important. Big particles, and if they aggregate, do not do well in vivo animal models. You'll get acute toxicity right away. So knowing that up front really helps us to not see unexpected effects that are not good. And then, even looking forward, you should be using vehicles that are optimal to go into patients. But again, if you're in proof of concept, you might need to use a small amount of solvent, and so we can do that. But those are some of the things that we need to know. Can you reproduce your formulation? Is it in a vehicle that's compliant for patients exactly, and that such will be compliant, like good for use in animals. So that's the start, and we can help with some of that. We have helped with that in the past. I could put up some manuscripts where we're quite involved. So that's what comes to mind right now.
In the Nanomedicine Innovation Network, our investment strategy in projects has been to provide support to projects that have commercial potential. Some may be very early stage, and of course, these are the types of projects, I guess, these are the sort of technologies and discoveries that the PharmaCore deals with. Very early stage therapeutic opportunities that haven't quite firmed up their all of their features and their value proposition and their competitiveness in the marketplace yet, and that's why they need your service. We all know about that commercialization valley of death where the technologies are very high risk, and as a result, private money doesn't want to necessarily invest because they don't see whether it's very risky whether they'll get any return on that investment. So can you give us any sense of, if people have a therapeutic discovery or a technology or a strategy with something they've been working with and they work with you, what's the value added for PharmaCore, from PharmaCore, for these early stage technologies and in relation to their positioning and progress along that commercialization pathway to market?
I think, so to answer that, Christine did kind of indicate, and so I'm just going to add to that we do have a lot of expertise in our team. Marcel has commercialized various products. So you do have access to us to manufacturing. Dr. Brent Sutherland, who's been doing that, and Marcel's also aware, and just having that other set of eyes is very helpful and getting you through. We also do suggest you know, we can set these up, getting clinicians on board, and we have links to oncologists, and that is also a good set of eyes to look at your project. And then having an early discussion with Health Canada if you've got some really good preclinical that can help guide you to finish up your GLP toxicology. They guide you, they don't tell you what to do, and you're first in human clinical studies, but getting that clinician on board, which we can do the introductions, is very important. They provide you with the information of the unmet medical need, and so forth. So that's another set of expertise that's needed early on. But yeah, we've seen some pitfalls, and so we can help people guide, and some people come to us and don't even know where they're going. So even just an open discussion helps people finalize what they want to do and then how we can get there.